Hello class, Mr. Roth here. Today's lesson is going to be on gene regulation. And this image that's on this first slide here should look familiar from our last discussion, which was about cell differentiation. And I want you to pay attention to the two scenarios. On the top you see a scenario where the gene is being blocked by the green repressor molecule. The yellow molecule would be happy to go forward and transcribe this particular gene. However, in this case, it is being blocked by the repressor. You can look at the other scenario here, and there's an interaction between some mystery molecule binding to the repressor, which causes it to release. The releasing of the repressor allows for the RNA polymerase to move forward and express the gene. So we're looking at a scenario here where the gene is off, and we're looking at the opposite scenario here where the gene is on. What causes one to be on and one to be off is based on environmental circumstances. If you look at the environment around this gene, we see that there are no mystery white molecules. The environment here, however, is different, and that ultimately is what is determining whether or not the gene will be on or off. So let's move forward. I also wanted to review something else that we talked about before, because it turns out that gene regulation is what allows for cell differentiation. What allows for a bunch of cells that are identical to become so radically different from one another in their structure and function, what allows for that is gene regulation. Certain genes being turned on, certain genes being turned off has dramatic effects on what the cells become. Okay, one last tie-in to something else that we learned before is just the central dogma. Where the genes are actually being turned on and off happens in here inside the nucleus, because we know that it's happening with the DNA. But there's a lot of effects downstream that ultimately are going to have the big effect on the different shapes of the cells. And so I just wanted to highlight that it's these proteins that are responsible for the big change in the shape and the function of our cells, because we know that proteins are in charge of most of that. Okay, so the first example that I'm going to talk about in regard to gene regulation is the lac operon. It is a very well understood, and it's kind of your classic example of gene regulation. And what it has to do with is it has to do with the breakdown of a sugar called lactose. Lactose is a disaccharide, meaning it is made of two sugars connected together, and here's an image of it right here. And for our bodies to properly process this type of sugar and bring it in and do cell respiration with it, it has to first be broken down. And so we have an enzyme called lactase that is designed to do such a thing. So if we look here, lactase is shaped just right to fit the lactose molecule. We call this little shape here the active site of the enzyme. And so when lactose, this disaccharide, binds, the enzyme will help to break it apart into monosaccharides. And so we don't always need this lactase enzyme around. In fact, in some parts of the world, there are adults who don't even produce lactase anymore. And the reason for this is because they don't drink milk. They don't actually have lactose in their cells that needs to be processed. And so, although they contain the gene that can make lactase, that gene for those adults is just turned off. There's no need to produce this enzyme for them. So I want to mention, let's assume that this gene here is the gene which codes for the lactase enzyme. And this is an analogous, these are both the same gene. But there are two scenarios. There's the scenario where the gene is off, and there's the scenario where the gene is on. I want to dive into why we would want to have it on or off. What's the point of having the possibility of switching it on or off? Well, one, if we have it on all the time, the cell is wasting not only matter, in the raw material it takes to build that enzyme, but the cell would also be wasting the energy that it takes to build it as well. So it's in the cell's best interest to only have the gene on when it's really necessary. And as we mentioned before, for an adult who does not have the lactose sugar in their diet, they're going to be in this scenario most of the time. Most of the time, this repressor will be blocking the gene and we will not make lactase. However, if you think of an infant who's consuming a lot of milk, the sugar that's in that milk is lactose, and so we're going to need the enzyme to be produced. We're going to want to be in this scenario for somebody who is consuming lactose sugar. And so just to kind of spoil how this mechanism works, I want to tell you that these little mystery white molecules are the sugars themselves. And so the sugar, we're going to soon find out, is the trigger for turning the gene on. However, this image here is a little bit more simple than how it actually works. And so I'm going to pull up a simulator here that should give you a pretty good view at how it works. So I've got to get this within our window here. Okay, it looks like it's visible. So what I first want to show you is I want to get you oriented with what the molecules represent. You'll see that I have a legend over here that's going to give you what they are. You notice that we have our DNA strand down here. It's a double helix, and certain regions of this DNA correspond to certain genes. So we know we have the LAC-I gene, which is located right here, and then there's another region of the DNA, which is what we call the LAC-Z gene. Pay attention to what the molecules are doing. You'll notice that this blue enzyme is binding to what we call the promoter region, and it is then going forward and transcribing that LAC-I gene. You'll notice that as it's transcribing, it's releasing a black strand here. The black strand is RNA, which is what we call a messenger molecule. It's kind of that intermediate step. You'll notice that this RNA will quickly turn into or be translated into a protein. And you'll notice the protein, if you look at the legend, is the LAC-I, which stands for inhibitor. And if you notice the behavior of that inhibitor, it goes and it likes to bind to this little region right before the gene. And most importantly, I'd like you to take note of its effect on the RNA polymerase. You'll notice that the blue RNA polymerase molecule cannot, it's trying to go forward right there, but it cannot because that repressor or inhibitor is blocking it. And so right now, the LAC-Z gene is turned off. Okay? We have not seen the LAC-Z gene be expressed yet, although the LAC-I gene is being expressed. Now there's a reason for this, and the reason is that the LAC-Z gene is what is responsible for producing our lactase enzyme. And this is the product up here. If you look at my cursor, this purple round enzyme is our lactase enzyme. It's responsible for breaking down lactose, which is this little disaccharide. And so I'm going to go ahead and inject some lactose. We're going to create a scenario where the person has just consumed a large glass of milk, let's say. So here you go. Here comes all the lactose. Here's all of those disaccharide sugars. And you'll notice immediately that they have an effect on the repressors. In fact, they remove the repressors, which allow the RNA to transcribe the LAC gene. And then you'll notice that that RNA becomes goes through translation and produces the lactase enzyme. So let me say that one more time. You'll notice that when the lactose sugars are present, they immediately bind to the repressors, making them inactive. In other words, causing them to pop off of the inhibitor site. Here it is, right there. It just popped off, which allows for polymerase to bind, move forward, produce the RNA that corresponds to the LAC-Z gene. That RNA then gets translated into the enzyme. And so here's our purple enzyme floating around. So let's pay attention to what the purple enzyme does. Purple enzyme, it appears to bind to those disaccharides and break them down into individual monosaccharides. And of course, that's what we mentioned before on the previous slides. We had mentioned that that lactase enzyme is what is responsible for digesting our milk sugar. Most importantly here, what I wanted to point out is that we did not see any of these enzymes around until that lactose became present. And that is the function of the mechanism. When lactose is present, LAC-Z gene is turned on. And this should make sense to you. If you really think about the mechanism, it should make sense to you that we want that enzyme around only when the sugar is present. Okay, class, that's it. I hope that this lesson makes sense. If you have any questions about it, please ask me. Or watch the video again. Sometimes a second watch will help you to understand. Okay, see you tomorrow.